Hello everyone. Let's start with our new question today. As you can see, this particular question was asked in GATE 2021. So, it is said that 0.5 ml of a bacteriophage. So, this representation that I have drawn here, this is not a scientific diagram. This is just a simple illustration to make you understand or to know what is a basic bacteriophage looks like. So, here we can see that this is a bacteriophage and this is a capsid head. This is called a capsid head, and this thing is a nucleic acid. This is a nucleic acid. This is a, we call it as a collar, okay, and this is a sheath, and these are called as the spikes, or you can call them as a tail fiber. These are like tails, so these are the tail fibers. Okay, so this is a basic structure of a bacteriophage. This is not a scientific diagram, like I just said. This is a simple illustration for you to understand. Okay, so this bacteriophage is taken from a huge stock. So they are saying 0.5 ml of an aliquot. Now, does anybody know what is an aliquot? What does aliquot mean? Okay. What is an aliquot? An aliquot is simply a sample. This is a sample that is taken from a huge portion. For example, there's a huge portion of these bacteriophages. Okay, there are huge portions of these. There are n number of bacteriophages. Okay, from this they have taken a sample. This sample is taken for what? It could be for a biological analysis or for a different kinds of treatments. So when a portion of this whole, a small portion of this whole is taken for any biological analysis or for other treatments, then it is referred to as an aliquot. This is a simple, you need to know the analogies because this is very important when it comes to exams like GATE. You need to understand each and every term properly, because if you don't know the basic meaning of an aliquot, then I think it is useless. Okay, you need to know the basic definition of each terms that are being used in the question, because in exams like GATE, these are very important. Now let us come back to the question. So 0.5 ml of an aliquot are used. Okay, 0.5 ml of an aliquot is used. This is having a concentration. What is the concentration? The concentration is 4 into 10 to the power of 9 phages per ml. Phages per ml, which means in 1 ml of this aliquot, in 1 ml of this aliquot, 4 into 10 to the power of 9 phages are present. Okay, this is simple understanding. Next, this is added to 0.5 ml of E. coli culture. Okay, they are saying 0.5 ml of this is added to an E. coli culture. Now let us see E. coli. Okay, E. coli is nothing but Escherichia coli, which is, let us simply, for your understanding, let us consider this as a E. coli. Okay, and this is having let us assume it is an E. coli. Okay, so this is having an outer membrane, and definitely there is a cytoplasm, cytoplasma, and there is an inner membrane. This is the inner membrane, and these projections are nothing but, what are these projections? This is nothing but a flagella. These are nothing but a flagella. Okay, 0.5 ml of, so now they are saying 0.5 ml of this is added to the E. coli. Now they are asking us what is the multiplicity of the infection. Okay, so to calculate this is just a basic understanding. I told you about the concept. Now let us solve the problem. Then what they've asked in the question, they have asked us multiplicity of infection. Let us all call it as multiplicity of infection as MOI. Okay. Now what does the term multiplicity of an infection mean? This simply means that it is a simple ratio. Understand that it is a very simple, it's a ratio. It's a ratio of what? It is the ratio of the attackers. Okay, let us call them as attackers versus what? Targets. Okay. So who are the attackers? Attackers are nothing but the bacteriophages, the viruses. Bacteriophage is nothing but it is a virus divided by, who are the targets? Targets are Escherichia coli, which is a bacteria. Simple. This is a simple concept. Okay. So who are attacking? Bacteriophages, which is a virus like we have just drawn. Okay. A virus is attacking who? A bacteria. Because E. coli is a bacteria. This is a simple way to remember this formula. So let us come back. Simply MOI is nothing but total number of phages. Total number of phages. One nothing but attackers divided by the total number of target cells. In our case, it is E. coli. Now, this is a simple question, simple formula that you need to remember. Okay. Next, first step we need to do is we need to find the total number of phages. We want this. Okay. So find it out. First, what they have given us? They have given us the concentration. They have given us the concentration of these phages. What is the concentration? 4 into 10 to the power of 9 phages are present in how much? 1 ml. So it is per milliliter. How much volume they have added? They have taken how much? Only 0.1 ml of this they have taken and they have added to the E. coli cells. Therefore, how do you calculate it? Total phages is nothing but concentration. Whatever they have given, just simply multiply it. Concentration into what is the volume. Okay. So 4 into 10 to the power of 9 per milliliter into 0.1 milliliter. So the answer is same. Same? Not same because it is 0.1. So you shift the digit. So it will be 10 to the power of 8 per ml. ml get cancelled. So it is just phages. So how many number of phages? Totally there are 4 into 10 to the power of 8 phages. First thing you need to do. What next? So what next? You need to find out the target cells. Okay. So B, the second thing you need to do is, okay, second thing you need to do is same. They have given us the, what they have given us? They have given us the concentration. They have given us the concentration, which is 2 into 10 to the power of 8 cells per ml per milliliter. And what is the volume? Volume they have used is 0.5 ml. So same like the previous one. What is the total cells? Total cells is nothing but concentration into volume. Concentration into the volume. So 2 into 10 to the power of 8 cells per ml into 0.5 ml is nothing but 1 into 10 to the power of 8 cells. Okay, so this is the total number of cells. Now we know total number of phages and we also know total number of cells. Now simply calculate the MOI. C is what? MOI. So what is the total number of phages? 4 into 10 to the power of 8 divided by 1 into 10 to the power of 8. So finally what we get is 10 to the power of 8 and 8 will get cancelled. So what is 4 by 1? It is simply 4. So the multiplicity of infection is simply 4. This is the final answer. If you know the formula, okay, this you can solve this in less than 2 minutes. I have explained it because you need to understand each and every term properly. If you understand it in the first place, it is very easy to solve. If I give you the same question again, you will solve it in 2 minutes. So this is the basic concept. I hope this is clear. Thank you so much for watching. Please do like, share and subscribe for more such GATE questions. Thank you so much.